Hi, my name is Emily Leonard, and I am a senior biology and public health dual degree student at the University of Massachusetts Amherst, and I'm also a member of the ICONS program here at UMass. And today I'm going to be giving a short description of my honors thesis research, looking at the effect of a synthetic antioxidant on zebrafish growth parameters and organ development. So what is TBHQ? TBHQ is a synthetic antioxidant that's commonly found in a lot of ultra-processed foods, from processed cereals to Little Debbie snacks to Cheez-Its. Honestly, a lot of the stuff that we really enjoy to eat contains TBHQ. And the molecular structure is right here for you to see. And the World Health Organization in 1999 determined that the average American can reach up to 90% of the acceptable daily intake value set by the FDA for TBHQ. And high-fat diet consumers can actually exceed the acceptable daily intake value by 80%. And a Tufts University researcher actually found that consumption since 1999 has actually increased 5% since 1999. And so this brings me to, well, we know that people are exceeding the acceptable daily intake, but what are the harmful effects of such consumption? And my research aims to answer that question. So previous research on TBHQ, people have actually found that in the short term, TBHQ can actually produce chemoprotective properties. And this could be due to its interaction with the NRF2 antioxidant pathway. So under normal conditions, the transcription factor NRF2 is bound to KEAP1, the protein KEAP1. And when it's bound, KEAP1 sends NRF2 to degradation. But under oxidative stress and reactive oxygen species, KEAP1 releases NRF2, and NRF2 is able to go into the nucleus where it turns on the transcription of antioxidant genes and deoxifying enzymes. So how does TBHQ play a role? TBHQ actually interacts with KEAP1 and prevents it from binding to NRF2. So NRF2 is always able to enter the nucleus and turn on these genes, making the cell think that it's always under chronic oxidative stress. And that's actually helpful when the body is fighting cancer, because it's constantly trying to fight these properties. In the long term, however, TBHQ has been shown to cause DNA damage and carcinogenic effects, and it's even been found to harm the immune system and reduce T cell activation in mice and humans. In zebrafish specific models, our lab has shown that TBHQ exposure is associated with decreased total length and a decrease in the number of beta cells at 72 hours post-fertilization. And as a little side note, our lab is very interested in beta cells and pancreatic islets because we investigate how environmental chemicals can be associated with chronic disease in the future, such as type 2 diabetes. And our lab has also shown that TBHQ exposure produces an increase in BioG and beta cells of NRF2A wild type zebrafish. And BioG is a glutathione analog used to detect oxidative stress. So it's showing us that TBHQ is in fact turning on this pathway that regulates oxidative stress. And we also see a decrease in pancreatic islet size at 96 hours post-fertilization. So based on this previous research, we hypothesized that chronic dietary exposure to TBHQ will cause abnormal pancreas development and alter the gene expression in zebrafish. So our methods were to make a 5% TBHQ diet, and we did so by dissolving TBHQ in 96% ethanol and spraying it onto the normal zebrafish feed. And then our control was just the regular 96% ethanol sprayed feed. And then the NRF2A wild type and mutant larvae began feeding on this diet at 5 days post-fertilization for about 5 months. So at 20 weeks post-fertilization, length was measured, and 4 representative samples from each treatment group was selected for RNA isolation. And then the top 3 samples with the highest RNA levels were selected for RNA sequencing. And we did a similar exposure paradigm, but only for 10 days. And we fed this time transgenic zebrafish for insulin GFP in the beta cells so that we could see the pancreatic islets at 15 days post-fertilization. And we fed these zebrafish starting again at 5 days post-fertilization, but only for 10 days until 15 days post-fertilization. And then we assessed length and islet area. So what were our results? We saw that TBHQ exposure negatively affects survival in adult zebrafish at 20 weeks post-fertilization. We also see a genotypic result where the NRF2A mutants have much smaller survival than the NRF2A wild types in the TBHQ exposure, but we don't see a genotypic difference in our controls, which is what we want to see. At 15 days post-fertilization, we still see a treatment effect where TBHQ negatively affects survival, but we don't see that genotypic effect at 15 days. Now going to total body length, it's already been shown that body length was decreased at 72 hours post-fertilization, but we also see a decrease in length at 20 weeks post-fertilization, and we don't see a significant difference in the genotypes here. We just see a treatment effect with TBHQ exposed zebrafish having a much smaller body length. And we do see a treatment effect in the wild types at 15 days post-fertilization, but we don't see it in the mutants, suggesting that maybe NRF2 might have, these effects might be NRF2 dependent, but we don't know for sure. And total body length, as you can see here, these are just some representative images of my controls at the top, my wild type and my mutants, and then my TBHQ exposed fish, and you can see that they are much smaller here. So we also wanted to look at islet morphology, as you can see here, and right here in part C is what a pancreas islet looks like under the microscope. And so we just took the area around that to see how large the islets were at 15 days post-fertilization. And we wanted to see if the area of the islets corresponded to the length of the total body. And as you would expect, as the zebrafish grows and has a longer body length, it's going to have a greater islet area. And we wanted to make sure that we controlled for this. So we normalized the islet area to the body length. And as we see here, there's a genotypic difference between the ethanol and the TBHQ zebrafish, but we don't see a treatment specific effect. So our hypothesis was incorrect in that it would affect pancreatic islet morphology. But going back to the RNA sequencing, we assessed RNA sequencing from the TBHQ as well as the controls. And as you can see here, they are clustering different, but we do have 2 outliers in our controls, and they are not clustering. They are not clustering with any of the TBHQ or the other ethanol exposed zebrafish. And we found that these are the only female samples in any of these samples. So we think that there might be a sex differential. There might be sex differences in the RNA sequencing. So these are just my preliminary results because we're still figuring out how we want to deal with these outliers. And as you can see here from the comparison with the ethanol wild type to the TBHQ wild type, there were 13 differentially expressed genes. And in the comparison between the treatments in the mutant genotype, there were 45 differentially expressed genes. And then when we compared the genotypes in the TBHQ treatment, there were 2,323 differentially expressed genes, showing that there is a large genotypic effect here. But specifically, I want to point your attention to, with the time that we have now, to the decrease in growth hormone. So growth hormone is involved in the positive regulation of growth, and the downregulation in the wild types makes sense because the TBHQ zebrafish were much smaller than the ethanol. But we don't see that going on in the mutants. So we wanted to see what mutant and wild type TBHQ exposed zebrafish had in common. And we see here that they have 3 differentially expressed genes in common. And I want to point your attention to this top one specifically. And this gene has been associated with the mouse phenotype of increased body weight. And we see that it's actually increased in the mutants and decreased in the wild type, suggesting that we would see a genotypic difference, but we don't actually see a genotypic difference in the wild types or the mutants. So we don't really know what exactly is going on here, and the RNA sequencing doesn't seem to be explaining our phenotypic results. But again, once we decide what to do with those outliers, I think that we'll see more promising results. So going through the key findings, we've already determined that survival decreases with TBHQ exposure at both 15 days post-fertilization and 20 weeks post-fertilization. At 20 weeks post-fertilization, however, survival is significantly different between the NRF2A genotypes, suggesting that NRF2A may have these effects might be NRF2A dependent. With NRF2A mutant zebrafish having the lowest survival. We've also shown that chronic TBHQ exposure negatively affects the body length of the zebrafish, both at 15 days post-fertilization and 20 weeks post-fertilization. However, these are more profound in the longer exposures. And we didn't find any significant effect on the area of the primary pancreas islets with this exposure paradigm. So this part of our hypothesis was proved to be incorrect. However, we do see gene expression differences between the exposures. So my future directions are to do more analyses on the RNA sequencing data, and in order to investigate the role NRF2 has on gene expression in TBHQ exposed zebrafish. I will also be doing oil red O staining to assess the changes in lipid accumulation in the liver at 15-day post-fertilization. And I'll also be doing nutrient analyses on these 15-day zebrafish to assess if TBHQ has effects on levels of protein, triglycerides, glucose, and cholesterol. So I just want to say thank you to my lab for all of their help throughout this whole process. And I would also like to thank Dr. Ravi Rajan for his help with RNA sequencing at the UMass Genomics Core facility. And I'd also like to thank my funders, the Commonwealth Honors College, as well as the National Institute of Environmental Health Sciences through Alicia Toomey-Larragee. Here are my references. And thank you so much. If you have any questions, feel free to email me. My email is right here. And I look forward to seeing you at the question and answer. Thank you.